This is the first set of notes on biotechnology. We'll be spending the next few weeks talking about various kinds of techniques in biotechnology and what they're used for, and we'll be doing some activities in class that reinforce this while we're also preparing for the EOC. So just a definition to begin with, biotechnology is the manipulation of organisms or their components to make useful products. This includes lots of different things, including some things that have been happening on Earth for thousands of years. Selective breeding of crops and livestock have been going on since civilization began. Using microbes to make wine and cheese, not at all uncommon for a long time. But now we've incorporated using DNA technology to make useful products in addition to these standard techniques. And so we're going to talk about some of the more modern things in addition to some of the older ones. One of the first things that happened back in the 1980s, 1990s was that scientists were trying to clone animals, and they knew they could clone animals from fetal cells, from embryonic cells, but some scientists felt like they could probably manage to make it happen from using a nucleus from an adult cell. And so what they did, these scientists in Scotland worked with sheep, and they took a sheep and they took a cell from the sheep's udder, which is the mammary gland of the sheep. Okay, that's going to be the donor nucleus for the clone. They took an egg cell from a second sheep and removed the nucleus from it. They fused the donor nucleus into the egg cell, and then they had a fused cell that was now the cell that was capable of developing into an embryo. Remember, the egg cell has everything necessary for the embryo except for that full set of chromosomes. Once you put the donor nucleus in there, it's diploid rather than haploid, then it has a full set of chromosomes and can begin dividing. So it begins dividing normally and develops into an embryo, which can then be placed in the uterus of a foster mother and develop normally into a lamb. This lamb they call Dolly, named after Dolly Parton, who is famous for her mammary glands because the original cell came from a mammary gland, is genetically identical to the donor sheep, the genetically identical to the sheep from which the original nucleus came that made the cell. This was a big breakthrough in biotechnology back in the 1980s and 1990s. And lots of animals have been cloned since then. So that was a big breakthrough as far as biotechnology went. There are a number of other techniques that are used for various kinds of things. We're going to talk about some of those. All of these things involve something called genetic engineering, which is manipulating the genes for some kind of purpose to make some product. One thing that's very commonly done now is gene cloning. And this uses plasmids from bacteria that you have put desired genes into to make multiple copies of a gene, and then and that will allow the bacteria to actually produce proteins from some other species. What we're making here is something called recombinant DNA. In doing so, we're joining the DNA sequence from two different sources. One source has the gene that we want to have a copy of, and the other source is the gene carrier or vector. And very often in biotechnology and genetic engineering, plasmids from bacteria are often used as vectors. Oftentimes the bacteria that is used is E. coli because E. coli is around all the time. It's ubiquitous. It's used for lots of things, and there are a number of different laboratory strains of it that are very safe. Now, here are the steps that you would need to follow in cloning some genes. First of all, you would need to isolate the plasmid DNA and also isolate the DNA that has the gene that you want to clone. You're going to treat both pieces of DNA with chemicals called restriction enzymes. These are special enzymes from bacterial cells that cut the DNA into fragments at certain places. Those are places called restriction sites, and they look for certain sequences of nucleotides and cut the DNA in a certain way, and we'll talk about how that's done. Then you're going to mix the plasmid DNA and your target DNA together and form what's called a recombinant plasmid. The recombinant plasmid is going to have the original bacterial plasmid plus the gene that you're trying to make copies of. Once you get that recombinant plasmid made, then it can be taken up by a bacterial cell in a process called transformation. And then the bacterial cell, as you grow it in culture, will form clones, exact copies, genetically identical copies from that original bacterial cell with that clone gene in it. and that gene will be replicated as many times as the bacterial cell is. These restriction enzymes are what we use. We call them sometimes molecular scissors because they're really used to cut and paste DNA. Each enzyme binds to DNA at a certain restriction site dependent on what that particular restriction enzyme is. And there are a lot of different ones, and a lot of them actually make something called a staggered cut that produces sticky ends. What these are ends of DNA that are single paired. They don't have a partner. They're single sequence, and they'll easily match up with complementary sticky ends and join together to form recombinant DNA. And the thing that sticks them together, it holds everything together, is DNA ligase. That's going to be our tape or our paste. Here's the process that you have. You have your E. coli bacterium with a plasmid that you can remove, and you have the cell that has the DNA with the gene that you're interested in cloning. So you'll isolate both of those, cut both of them with the same restriction enzyme. Notice the one that's used here is one that produces those single-stranded sticky ends. We're going to put the plasmid and the targeted gene in the same culture, and they will match up just fine like that. And then we're going to have this recombinant DNA plasmid after it's been treated with DNA ligase, which will join those molecules together. And here's our gene that we want to clone. Now we're going to put this recombinant DNA plasmid into a bacterium, and then as the bacterium reproduces, each daughter cell will have that gene that we're interested in cloning. The genes can sometimes be inserted into other organisms as a method of gene therapy, or the proteins that can be produced by this gene can be harvested and used directly. One thing that is very commonly used is bacterial growth of various kinds of hormones from human hormones that are difficult to come by. One is human insulin. For many years, people who were diabetic had to take pig insulin, which was very similar to human but not identical, because it was much easier to get insulin from pigs than it was from humans. After all, humans need their own insulin, and there's not really a ready source of cells that make insulin. But now we can take the human gene for insulin and put it into a bacterial plasmid, grow bacteria in huge cultures, and produce lots and lots of insulin relatively cheaply. And it's also human insulin, so it's identical to what you need for your cells and works better than any other kind of mammalian insulin would do. There are a number of other hormones that are used this way. Human growth hormone is another one that's produced this way. And there are other kinds of proteins and other products that can be made in similar fashion. When we have transgenic organisms, we are making something called genetically modified organisms. They have at least one gene from another species, and there are a number of different things that are available now. There are plants that are being produced that are more resistant to pesticides and pests. There are plants and animals that are made that provide nutrients that help address various kinds of malnutrition needs. There are cows that actually produce several kinds of human hormones in their milk. And so this is it's kind of a brave new world of genetically modified and biotechnology that an advance that's from what's been happening in the thousands of years in the past. Now, there are concerns about genetically modified organisms. Part of it has to do with the concerns that you might accidentally introduce some allergens into the food supply that could cause major problems for those, particularly people that have very numerous specific food allergies. Another concern is the spread of genes to closely related organisms. If you have a gene in a plant that makes it resistant to pesticides, and that gene gets into a different plant that's maybe a weed, it might be difficult to get rid of those weeds. There are a number of concerns about these that are being addressed by regulatory agencies like the FDA and places like that. And so they're talking about how to address the safety of it, how to label the things like that, and how to safely use biotechnology. I'm now going to show you a video about a story about a particular kind of genetically modified salmon.
For the first time, the government is considering whether to allow the sale of food from a genetically altered animal, the FDA taking it up today. A company has tinkered with the genetic material of salmon so the fish can grow faster and reach groceries sooner. Are eaters ready? Here's Lisa Stark.
It looks like any other salmon, but opponents call it Frankenfish and hope to keep it off your dinner plate. Here's the difference. Both these fish are 18 months old, but the larger genetically engineered Aqua Advantage salmon grows twice as fast as the regular salmon.
This is the first genetically engineered animal that would be introduced to the food supply. So it's a big deal.
Atlantic salmon grows in fits and starts, but scientists found that adding genetic material from a Pacific salmon and an eel-like fish helps them grow round the clock.
This fish is indistinguishable from the traditional foods.
How can you say it's indistinguishable, though, when it clearly has a different genetic makeup than an Atlantic salmon?
But it tastes the same, it looks the same, and the biology is exactly the same.
FDA scientists today agreed.
Aqua Advantage salmon is Atlantic salmon, and food from Aqua Advantage salmon is as safe as food from other Atlantic salmon.
Still, critics claim more studies are needed on how the fish might affect humans and the environment. For instance, what happens if these altered fish somehow make it out into the wild? Would they breed with native salmon? Would they crowd out the wild fish? The company insists there are safeguards.
They're all sterile, and they're raised in physically contained facilities.
But opponents say there's no guarantee all the fish will be sterile. And there's already one case study in how the best laid plans can go awry. Canola plants used to make canola oil were genetically engineered to be resistant to weed killer. The problem, their seeds have now migrated from the company farms, carried by the wind, and perhaps by trucks taking them to market. They're now growing on the roadsides of North Dakota. Scientists worry they may create a hard-to-kill superweed. As for the genetically altered salmon, one question still on the table: will folks eat it? And will they even know what they're eating? Because the FDA has to decide if the fish will even carry a special label. Lisa Stark, ABC News, Washington.
Now, another goal of genetic engineering these days is something called gene therapy. This is an attempt to treat a disease that is caused by a gene that does not work properly. It would require altering the genes of the person who has the condition, and it attempts to treat that disease in a way to make it curable. It would require delivering the gene to a cell type of a patient that has been grown in culture via a retrovirus, which is an RNA virus, and then grow those cells, the gene therapy cells in culture, and then return them to the patient. This works best if you're trying to treat a condition that you know is caused by one particular kind of gene. And so it has to be something that has a single gene rather than a multiple or polygenic trait. Here we have a clone gene from a normal allele into, we take the RNA version of it and put it into a retrovirus. Remember, retroviruses are RNA, and they have an enzyme called reverse transcriptase that allows them to be changed into DNA. And so you would put the viruses infected into the cells that you were trying to treat the patient with, and then if you put that in there, then it will become part of those cells. You grow the cells in culture and return them to the patient, hopefully enabling you to cure the condition. This is the last part, or this is the end of this particular set of notes. There will be more notes coming about some other genetic engineering and biotechnology topics.